condition in which the inducer is present, and it is in switched on condition. So in switched on condition, what the inducer of where we have various segments. This is P, promoter gene, this is repressor, this has to be made. Then we have operator, and then we have the three structural genes Z, Y, and A. Z, Y, and A. So to this attaches the RNA polymerase. RNA polymerase attaches with this, and as a result, this activates, and a messenger RNA as a result of transcription is formed, which ultimately, as a result of translation, and this is as a result of transcription. Translation here, a repressor protein is formed. Repressor protein is like this. Inducer is present here. Inducer. The inducer in this case is lactose. Lactose is the inducer. So this has to enter in. So right now, see, no synthesis has taken place. No synthesis of trans-acetylase, no synthesis of beta-galactosidase, no synthesis of permease. Permease, which is secreted by Y gene, allows the entry of inducer into it. So right now, nothing has been secreted. So always there should be little permease in here. Little permease should always be in the cell. So always little permeable. Now here the repressor protein is synthesized, but this repressor protein, where does it bind? I told you that it binds at the operator gene. So this repressor binds with operator gene. Now, as I have told you here, when it binds, then what happens? The second RNA polymerase, which is attached to this P, RNA polymerase, which is attached to this P, it cannot slide over this gene. It cannot slide over. Usually when this is not present, it slides over it. And then 3 enzymes are produced. But right now there is a so this RNA polymerase is sized. Now what happens when an inducer, which is lactose, when it enters in, this repressor protein which has gone from here, this is the repressor protein which has gone here. Okay, this repressor protein, it has more affinity for this than to O. That means repressor protein, I'm just making the structure, repressor protein has more affinity for inducer. And what is the inducer here? Inducer here is lactose molecule. It has more affinity for lactose molecule than operator. So then O means operator. So it has more affinity for inducer than operator. As a result, what will happen? It will leave this because it has more affinity for this. It will leave the site O. It has left the site O now, and it will attach to the inducer. It has attached to the inducer. Now see, it has attached to the inducer. So this is a complex formed. What is the name of this complex? This is inducer repressor complex. or lactose repressor protein complex. This complex has been formed. Now, see, there is a change in the shape of this complex. Here we have this repressor protein, inducer with this repressor protein. Now, because of this shape, it cannot bind. Because of this shape, here I'm writing that permease should always be there. Little amount of permease should always be there to allow the entry of lactose in. And now this, because of its shape, this lactose protein complex cannot bind. This cannot bind at site O. It cannot bind at site O because of the change in shape. The shape has changed, the structure has changed, so it cannot bind at site O. When it will not bind here, what will happen? This RNA polymerase, which is here, there is no hindrance, and it will slide over this. And when it slides, RNA polymerase slides over this structural gene. This is structural gene. When this slides over this structural gene, transcription will take place. What will take place? Transcription. And as a result of transcription, what will be formed? Messenger RNA. As a result of transcription, messenger RNA is formed. And now what will happen? This messenger RNA will undergo translation. This will undergo translation. And as a result of translation, 3 enzymes will be formed. And which enzymes? Z will form beta galactosidase. Beta galactosidase. Y will form permease. And A will form transacetylase. And they will perform their function. So this is switched on condition of lack of operon. Switched on condition of lack of operon. When it is on, the repressor protein cannot bind at A site because of its change in shape and size. Because it forms lactose repressor protein complex, it cannot bind to O site. So what happens? This RNA polymerase, which is at the P site, it slides over the structural gene, results in transcription, formation of messenger RNA, and finally formation of the proteins, which is in the form of enzymes here. And all the 3 enzymes are formed. Then they carry out their function. What is the function of this? This is A, I'm writing this. This is B, and this is C. What is the function of A? Beta galactosidase converts A, which is beta galactosidase, converts lactose into, lactose is converted into glucose and galactose. Function of B allows entry of, lactose in the cell, in the prokaryotic cell or bacterial cell. And what is the function of C? C metabolizes the effect of the toxic thionucleosides, which gains entry with the lactose, which also come inside by chance with lactose. So their toxic effect is metabolized. So this switched on condition of lack of operon is clear to you all? Children, this switched on condition of lack of operon is clear to you all? Yes. This is outside the cell, and this I have made the bacterial cell. What is happening inside this bacterial cell? Is it clear, beta? Yes, Paras? Paras is saying it's clear. I want other responses also. Sukh Sahaj, okay, beta. Then, very important question. Many a time the questions, the answer has been asked. Yes, bache, is it clear? I have got just two responses, Paras and Sukh Sahaj. I want others also to let me know if they have understood the switched on and switched off condition of lac operon. Okay, Parthavi, I've got your response. Yes. Amit Katoch. Okay, Gunjan. Karthik Thakur. Mariam, Amit Katoch. Okay. Girish. Sunidhi Chitra. Girish. Arif Wani. Okay, Aditi. See, you need to prepare this question properly. Chapters are important. These chapters, like biotechnology, both the chapters, this chapter, what do we, this chapter, molecular basis, these all chapters are very important. Which are always in switched on condition and the ones which are always in switched off condition. And whenever required, they can be switched on, or the ones which are always in switched off, they can be switched on. So the one which we have done right now, that was lac operon, that is an inducer operon. So inducible operons are the ones which are mostly in off condition, but they can be switched on. So operons, as I told you, are of two types: inducible, which are most always in switched off condition, but can be switched on. Switched on when required. And this, the example of this was, which we did lac operon. So lac operon, in which we saw that mostly it is in off condition, but whenever there is the lactose molecule present outside the cell and it is required by the cell, it has to go in, it goes in, then it comes into switched on state. Now there are some operons which are also known as repressible operons. Repressible operons, and these are the ones which are always in switched on, always opposite of this, always in on condition, but can be switched off, can be switched off. Example of this is tryptophan. Tryptophan operon. Tryptophan is an amino acid, and this amino acid is continuously required by the body. So we will do this repressible operon, which is most of the time in on condition, but can be switched off when required. So this we have done this. Now we are onto this. So tryptophan operon, tryptophan. Basically, this tryptophan is an amino acid, and this amino acid tryptophan is continuously required by the body. Continuously required by the body. That means continuous synthesis of tryptophan is required. This is a very essential amino acid and is continuously required by the body. That means all the time it has to be formed. And that is why the operon which is involved in the formation of tryptophan is always in switched on condition, but can be switched off when the body has sufficient oxygen. Now what happens? That means this tryptophan operon is a repressible operon. Repressible operon. So whenever it is always in on condition, and whenever we want, we can off it. So whenever we want, we can off it, but normally it isn't always in the switched on condition. Now, when we see the operon, how does it look like? So we have the same structure, same type of operon DNA segment. Here we have P, promoter site. Then we have R, the inhibitor site, which produces repressor protein. Then we have another P site. Then we have operator site. Then we have, in case of lac operon, we had structural gene with with 3 segments, but in this case we have 5 segments. The structural gene has 5 type of genes here. One, I'll make it a little bigger. 1, 2, 3, 4, and 5. The structural gene has 5 various types of gene here. Okay, now this is the structural gene. We can name it trypto AB, or we can name it trypto AB. In some cases, it is named as trypto AB, or we can name it ABCD. A, B, C, D, E. This also we can name it, and we can name it trypto ABA also. TRYP T or TRYPA. This also we can name. So these are the various segments of this operon. Now here P is the promoter. P is promoter gene. And again, like lac operon, it attaches to cyclic AMP for energy and RNA polymerase to continue the process of transcription and RNA polymerase. So it attaches to these 2 things, this P and this P. Both promoter genes, they attach to cyclic AMP and RNA polymerase. Then we have R. R is inhibitory gene. And this inhibitory gene, it produces a repressor protein, which binds at the O site, which binds at O site, operator site, which binds at O. Whatever it produces, it binds at O and doesn't allow the RNA polymerase to slide over it and to undergo the process of translation. Then we have O. This is operator gene. And this operator gene, what it does? Binds with the repressor protein to stop the sliding of RNA polymerase, which is attached here. Then that means it regulates the transcription. This will undergo transcription. This is regulated by this transcription transcription of structural gene, regulates the transcription of structural gene. This is the function of operator gene. Then we have structural gene. This undergoes transcription and translation to form enzymes. Okay, and which enzymes are formed by ABCDE? All A, B, C, D, E, all the genes, they produce enzyme, they produce enzyme tryptophan synthetase. They produce the enzyme tryptophan synthetase. A, B, C, D, all they produce the enzyme tryptophan synthetase. And what is the function of this tryptophan synthetase? Tryptophan synthetase converts the raw material into tryptophan. That means raw material, whatever is raw material there, this raw material under the action of this tryptophan synthetase, this enzyme under the action of this tryptophan synthetase gets converted into tryptophan, which is an amino acid. Gets converted into tryptophan, which is a very essential amino acid. I told you this is an essential amino acid, continuously required by the body. So this converts raw material into tryptophan. Now, one more important thing before I start with the functioning of this operon, I want to tell you that cyclic AMP along with RNA polymerase, RNA poly, I'm writing this attaches to this P, and then it brings about the transcription. This RNA polymerase slides over this R gene and it forms the transcription, after transcription messenger RNA and then repressor protein. Same way, another CAMP and RNA polymerase is attached to this P. And when this is attached and there is no hindrance at the operator side, this RNA polymerase slides over this and produces tryptophan synthetase. Is it clear, children? This is clear? Till here, this is clear? I'll make you understand few more things. Yes, beta? Is it clear? Anything you want me to repeat? Yes, bache? Okay. Okay. Sukhsahaj Vanshika Paras. Okay. So, okay. So I've got some replies, and now I'll tell you something more about this. What I want to tell you now is, yeah, when there is no hindrance, this will bind over, this RNA polymerase will slide over this structural gene, and as a result of sliding over this structural gene, transcription will take place, and because of transcription, tryptophan synthetase will be produced. And this tryptophan synthetase, what it will do, it will convert the raw material in the presence of this tryptophan synthetase will get converted into tryptophan, which is an essential amino acid. Okay, children, is it clear? This introduction part is clear? Switched on and switched off, I'll start tomorrow, because right now if I start, I'll not be able to finish with it. So this tryptophan operon, which is a repressible operon, which is all the time switched on, but when we need, it can be switched off. So this operon is clear, beta. The introductory part is clear. This was the introduction. Tomorrow I'll start with the working of tryptophan operon, and then we will continue with other topics. But lac operon is also clear, beta. Any question from the lac operon part? Any question from the lac operon part? Yes, bache? From the lac operon part, any query? Any query from the lac operon part or tryptophan till here, the introduction part? Yes, bache, any query, any question? Everything is clear, Sukh Sahaj is saying. Okay, bache. Yes, to others also. To others also. Mitali, Sunidhi, Mansi, Mariam, Vanithi, Jasmine, Shruti Lakde, Navneet, Vanshika Singh has answered. Aditi, Gunjan, Chirag, Girish, Amit Katoch, Karthik, Parthivi, Sheetal, Simran, Sukhman, Suzanne, Tanish, Vanshika, Karthik Thakur. Anything in this which you could not understand? Tryptophan operon or lac operon? Yes, beta. Okay. So the time is over. I'll end the meeting. Tomorrow we shall start with this, with the functioning of tryptophan operon, switched on and switched off condition. Until then, just stay fit, stay safe, and take care of yourself. Okay? Okay, beta.